Workshop 2021 of AR Conference. In this break room, we will discuss how to prepare files for uploading to gEAR and how to curate a plot using gEAR. This is Yang. My colleague Elena will co-host this section with me. Before we start, please go to the website umgear.org/workshop to download the demo files that we are going to use for today's practice. When you log into that page, you're going to see multiple downloading links. We are in break room 4, dataset uploading and curation. There are multiple demo files that I prepared for today's purpose. Due to the time limitation for this section, first I will discuss some of the main points related to preparing the files to upload. Following that, I will give a short demonstration about how to curate a static Disney plot or a Violin plot. If you are interested in other curation options, such as bar plot, please refer to the rest of this PowerPoint. Let's go back to the data preparation. If your goal is to visualize your expression profile in gEAR, there are two options. Option 1, you can prepare 3 type files named as expression.tab, observations.tab, and genes.tab with a metadata Excel sheet, which include a summary of your experiment. Alternatively, you can prepare 1 Excel files which include 3 separate sheets, expression, observations, and genes, along with the metadata file. This option will be quite challenging if you are preparing the files for single-cell RNA-seq due to the large dimension of the expression matrix. If you plan to use the single-cell workbench in the gEAR, the format requirement will be slightly different. You will still need 1 metadata Excel sheet along with 3 type format files, barcode TSV file, genes TSV, and sparse matrix. I will give a detailed explanation in the following slides. First of all, let's talk about the metadata file. You can download the metadata file directly from the gEAR website. This metadata sheet is mainly used to describe your experiment, and it is important to note that not all the fields are mandatory. Only the rows which are tagged in Y are required to be completed. There are a couple of things I want to emphasize. First, the annotation release number must be a numeric values. Secondly, if you would like to provide the GIC number, please make sure it is publicly available, otherwise leave it blank. And as long as the GIC number is filled in, the infos for the following rows will be extracted from gEAR website automatically. If any of the mandatory field is left unfilled, gEAR validation script will highlight the missing part in red. Next, I want to discuss how to prepare the Excel sheet for uploading. In the Excel sheet, there should be 3 tabs: Expression tab, Genes tab, and Observation tab. In the Genes tab, make sure the first column is keyed on ensemble ID, followed by gene symbol. In the Expression tab, the first column also required to be keyed on example ID, followed with the normalized values of your samples or your cell. For the Observation tab, the first column will be named as Observations, followed all the annotation infos you want to include it, such as treatment, genotype, time, and other variables. And one thing I want to emphasize, the names in the observation column must be identical to the column names in the expression sheet. Otherwise, the gEAR cannot merge the expression matrix with the observation matrix together. If you have any replicate column you want to include, you can fill in the replicate numbers corresponding to each sample. But make sure do not include the replicate column at all if you do not have any replicate. Next, I want to talk about how to prepare the 3 type files. Similarly as we see in the Excel format, instead of one single Excel file, the 3 type file should be separate. Expression.tab, observation.tab. In the expression.tab file, the first column also needs to be keyed on example ID. The observation tab file is similar as we observed in the Excel format. So the first column is observations, followed with all the infos you want to include it. If you have any replicate, include that also in the observation tab file. For the genes tab, make sure the first column is keyed on example ID, followed by gene symbols. For all these 3 files, you need to zip them together as a tar.gz format for uploading. If you are preparing the 3 type files for single-cell RNA-seq dataset, the overall requirement is the same as what I showed before. But for the observation tab, you can also include the t-SNE coordinate to duplicate the t-SNE plot that you generate by yourself. And also, you can include the cell type names in the observation tab. We highly recommend it limited the length of the cell type names less than 15 characters. This is due to display purpose. And if you want to display the same color code as you use in your manuscript, you can also include the corresponding hex code and named as, for example, cell type underline colors. In that case, gEAR will display the t-SNE plot with the same color code of your own version. If you are interested of using the primary analysis function in the single-cell workbench, you can add extra column named as cluster underline label. The gEAR will add the primary analysis function if you see the tag in the observation file. The requirement for the genes.tab is similar as we see before. Make sure keyed on example ID. Next, I want to discuss how to create files that you want to use the single cell workbench. For this purpose, you'll still need a metadata sheet along with 3 files, but these 3 files are slightly different than what we see in the Excel format or what you prepare for the RNA-seq. The 3 files are named as Mtx Files, which is sparse matrix, barcode TSV files, and genes.tsv files. The Mtx format include a headline which starts with a percentage symbol and followed by the number of rows, the number of columns, and the number of non-zeros in the matrix. The barcode TSV file includes a barcode for each cell, and the genes.tsv file must be keyed on ensemble ID. All these 3 files must be zipped together as tar file for uploading. If you still have questions about how to prepare the files for uploading, we have direct downloading links on the gEAR website. You can download the template and prepare the files correspondingly. That's all the major tips related to dataset preparation. In the next few minutes, I will use a demo single-cell RNA-seq dataset to upload to gEAR, followed by curation of a static t-SNP plot and a Violin plot. So let's go back to the main page of gEAR. In order to upload the dataset to gEAR, there are 2 options you can submit your data. First, if you click the triangle next to your account name, you're going to see a drop-down window which showing the options Upload Dataset, Gene Expression. You can click this link. Or you can look at the left bottom corner of gEAR website. There's an icon called Launch Dataset Uploader. Both ways can jump to the uploading page. Let's click Launch Dataset Uploader icon. This is a page. Step 1. Upload metadata file. You can see there's a download link to download the metadata template. Since we already prepared the metadata file, so we're going to upload directly. Click Upload Metadata File icon. Now go to the Download folder and select single-cell RNA process demo files. And in this folder, you're going to see a gEAR metadata demo Excel file. Select this file and open. In this page, you can see all the summary of all the info that you fill in the metadata sheet. If any required fields are missing, you will see a highlight in red. And for this demo file, everything is filled in the correct way. So we can continue to the next step. This next step is uploading the expression data. Over here, you can see the multiple download links corresponding to different format, Excel format, Mtx format, and 3-type format. You can download the template to prepare your own dataset. Let's select the prepared demo file for uploading. So over here, in the download folder, you're going to see a tar gangz file. This is the one that we're going to upload to gEAR. As I mentioned before, these files are prepared with 3 different type files, and all these 3 type files are zipped together in a tar.gz format for uploading. This is a single cell profile. It may take a few seconds to load to gEAR. When the uploading is done, you're going to see a summary of your expression matrix, the number of genes with the number of cells. Now we click Submit. Now we are in the curation page. Click Curate this dataset. And click the icon Add Display. Over here, you can see the Dataset tab. We'll leave the default option Primary Data. If any of the users using the single cell workbench did the analysis, you can select Stored Analysis to display the result from the single cell workbench. Since we're using our own dataset, we're just using the default Dataset tab, Primary Data. In the Display tab, click the drop-down window and choose Display Type. So for now, we're going to curate a t-SNE static plot. Next, we need to fill in a gene symbol which you're interested. For example, SOX2. For the display parameters, for the x-axis, we're going to select t-SNE1. For the y-axis, we're going to select t-SNE2. All these names are included in your observation tab. On the right side of the window, you're going to see expression t-SNE plot. There are more options you can polish this plot. Click Show Color as Legend. We can also colorize the legend by cell type. So we click the drop-down window and select cell type. In that case, you're going to see a side-by-side t-SNE. The left one is the t-SNE plot with the gene expression levels, and the right one is overall cell type names. And over here, this is a legend of all the cell type in your dataset. If you're unhappy with the order of legend, you can drag the names around. So on the left panel, you see the order of cell type. You can select any cell types you want to change the order. For example, inner hair cell, just drag them up and down. You can see the legend order is changed correspondingly. If you are okay with this display, provide a name for this display and save. Over here, you're going to see a thumbnail of the image that you just created. If you decide to put this one in the front page of your profile, make sure to click Make Default. If you're still unhappy with the plot that you generate, you can still edit by clicking the Edit buttons here. So now jump back to the display parameter that we used before. So let's click Show Color as Legend. Over here, you can see more options, for example, plot by group. If we select, plot by group with cell type, what it does basically generate the t-SNE plot per cell type, which you will align in one row side by side. it is kind of busy and tiny to see. So you can rearrange the display options. What you can do is you can put max columns per row to rearrange how many t-SNE plot you want to display per row. For example, we put 3 here. And then the t-SNE display will change with 3 t-SNE in one row with multiple columns and rows. So if you move to the end of this t-SNE plot, you can still see the expression plot of the genes of interest, followed with the t-SNE plot with all the different cell type names. And if you're okay with this display, just click Save Display. Now the t-SNE plot is changed to 3 t-SNE plots side by side per row. Next, I want to show you how to create a Violin plot. Click Add Display icon. And in the display type, choose Violin. And similarly, put a gene symbol based on your interest. And for the display parameter, the X, we're going to select cell type. And for the Y, we'll leave the default expression. And for the color code, we can color by cell type also. Now preview the chat. You can see there are Violin plots side by side for each of the cell type. Similar as what I displayed before, you can move the violins by dragging it individually up and down, so the order of the Violin plot will change. Also, if you are unhappy with the color code, you can click the color panels and choose the color that you like to use. So if you're okay with this display, put a name and save the display. Now you have 2 display options. One is t-SNE plot, another is Violin plot. You can decide which one you want to use as a default display in the front page of your profile by click Make Default icon. This is the demos for making the static t-SNE plot and Violin plot. If you are interested about other display options, please go through the PowerPoint that I displayed at the beginning. There are more demos for bar plot, line plot, dot plot. And also I want to emphasize, so if there are no t-SNE coordinate in your observation files, when you add the display, you may not see the option of t-SNE coordinate. So if you are trying to create a t-SNE plot, make sure you include the t-SNE coordinate in the observation tab. This is the major point I want to cover for today. And if you have any questions, feel free to email us and have a great day.